Hello and welcome to this Springboard video on cell signaling. I'm Adam, I'm a PhD student here in Oxford, and I study how our cells communicate in cancer and neurodegenerative diseases. Okay, so starting with the objective of this video, I firstly want us to understand the fundamental importance of cell signaling for all life on Earth. And then I want to go on to look at some specific examples of signaling systems to see how each signaling system is adapted for its specific job. And finally, I want us to think about what cell signaling can teach us in terms of human diseases, whether that's helping us treat them or even just helping us understand what's really going on. But okay, what is cell signaling as a start? Well, cell signaling is the process of cells using special chemical messages to communicate with each other or to respond to their environment. Now that might sound quite dense or overly complicated at first, but actually it's a concept that most of us will probably inherently understand. So for example, I think we all appreciate that our neurons, some of which we can see here in the top left, send electrical signals to each other. And it's those electrical signals which let us see, sense, touch, and respond to the world around us. And I think we can also all appreciate the effect that hormones can have, like testosterone, estrogen, insulin. Those are all individual signaling hormones, which let cells throughout our body coordinate really big responses, like developing our bodies through puberty, or controlling how much sugar there is in our blood. But okay, those are some really well-known examples. But actually, that's just the tip of the iceberg. Cell signaling is fundamental to life, and it will regulate essentially every process in biology in any cell at any given time. And we can feel that. So a lot of the sensations we have, like when you feel the need to take a breath, that's actually a cell signaling process. There are cells inside your body saying there's too much carbon dioxide in your blood, take another breath. And the same thing is true of something like a heartbeat. That's millions of cells coordinating their movements. You get one smooth heartbeat to drive blood all the way around the body. And even at the earliest stages of life, your cells need to coordinate so that they know where they are and what kind of cells they're meant to be. So cell signaling systems are responsible for making sure that your limbs grow in the right place, or that you develop your heart properly or your lungs. And that's at quite a large scale, but when we zoom in to the cellular level, again, every single system and every process in biology is usually regulated by some cell signaling mechanism. So that can include the fast, immediate responses like the electrical impulses sent by nerves, or coordinating the movement of this cell as it walks around. But it can also be slower, longer-term decisions like when cells should divide and multiply, or the exact opposite, when cells should undergo deliberate, organized cell death, called apoptosis. And of course, when we get into real living systems, we get combinations of many cell signaling processes, which can drive really complex events. So just as an example, here on the left, we can actually see a small ball of tumor cells. So if you look inside any of these cells, you'll find they have increased cell growth signaling pathways and decreased cell death pathways. And what that means is this small group of cells can eventually keep dividing and dividing to form a cancer that will grow and grow and really just won't stop. On the other hand, you can get events like this. So on the right, we see 3 cells on the outside, and these are part of our immune system. These are called T killer cells, and it's their job to find infected cells like this one in the middle and kill it so that the infection won't spread to the rest of the body. Now, how do they do that? Well, firstly, they have to massively multiply themselves, and then they have to move around the body tracking infections. And only then, when they found that, they need to be able to identify which cells are infected and which cells aren't, and then they need to force the infected cells to die, essentially. Um, that's a lot of processes, and each and every one of those steps will be controlled by at least one, usually many, cell signaling pathways. So cell signaling really can be life or death, and one of the best examples of this is adrenaline. Now, adrenaline you'll probably know, but it's the key driver of the fight or flight response, which is the feeling we all get when we're under stress. And the point of the fight or flight response is to prime us for a short but really intense bit of exercise that could save our lives. Now, one of the reasons that adrenaline is such a good example to look at is that it acts in the same way that a lot of hormone signaling pathways do. So adrenaline, shown here in yellow, is actually a small, simple protein molecule, like a lot of hormones are. And it travels around the body until it finds a cell that is displaying this receptor, shown in blue, on its cell surface. Now, this receptor only binds adrenaline. And when adrenaline binds to it, the receptor becomes activated. And when the receptor becomes activated, the cell that it's attached to becomes activated and starts working towards its part of the fight or flight response. Now, in different parts of the body, that causes different effects. So in the heart, that means we get an increased heart rate. More blood pumping means more oxygen to our muscles when we need to start running. Also, it changes how we sense the world. So our pupils dilate, we get tunnel vision, and at the same time that we're getting blood to most parts of our body, more blood to most parts of our body, we're actually cutting blood supply to the non-essential bits, like our digestive tract. So all of these different effects raise the question: what makes adrenaline work? How does adrenaline, as a signaling system, suit its job in having to set up the fight or flight response? I want you to take a minute here, pause the video if you want, to think, what traits will the adrenaline signaling system need to do its job well? Okay, we're back. So there's probably a lot of answers here, but there are three key points that I wanted to highlight. Firstly, adrenaline needs to affect the whole body. We saw that it has effects on the heart, on our eyes, on our senses, and our digestive tract, every part of the body. So it clearly needs to be able to reach everywhere in the body. Secondly, it needs to be able to do that fast because we don't have a lot of time to waste in a life or death situation. And finally, the adrenaline signaling system needs to be adaptable. And what I mean by that is, in every different part of the body that it reaches, it needs to create a different effect. And sometimes those effects are actually the exact opposite. So we said we'll get more blood supply to our muscles, to our arms and our legs, for example, but at the exact same time, the exact same molecule of adrenaline is able to cut blood supply to our gut. So how does it do that? Well, we'll work through these one at a time. Firstly, whole body signaling. How does adrenaline reach the whole body? Well, in the same way that any other hormone does, it gets put into the blood supply. Every cell in our body needs oxygen. And so every cell in our body is, pretty much at least, is directly connected to the blood supply system, our veins and our arteries. So adrenaline is released directly into the blood by specific endocrine cells. And endocrine just means these are cells which make and store hormones. And we can see, when hormones get delivered directly into the blood, they'll very quickly come across their target cells. They'll be able to move out of the blood and bind the receptors on that cell to activate it. Which brings us to speed, because all of this needs to happen really quickly in a life or death situation. So how does adrenaline act so quickly? Well, it's actually quite special in that regard, because adrenaline is directly linked up to our nervous system. So this image on the left, all of the bright regions shown are regions with a lot of neurons. And this bright speckled region is the adrenal gland, which produces and stores adrenaline. Now what that means is that within a couple of seconds of seeing or sensing a threat, our brain can have sent a signal to the adrenal gland saying, release adrenaline now. But The other half of that is that adrenaline needs to be ready to go. Producing a protein like adrenaline can take minutes or hours in a human cell. So it needs to be done beforehand so that we're not wasting time when it's needed. So how do cells do that to increase their speed? Well, they pre-package it. So this image on the right, the white boundary is a single cell, and on the inside are these dense hormone granules, which will be made of almost purely adrenaline. And very rapidly, they'll get this electrical signal from the nervous system, and these granules will quickly be dumped into the blood where they'll dissolve into all of their individual adrenaline molecules, meaning that we can almost instantly release a huge amount of adrenaline. Now, what are all the effects of this? Well, it takes us about 2 seconds from detecting a threat to get adrenaline into the blood, which is incredibly fast. And after that, the blood system itself will reach every part of our body within just 25 seconds. That means within around 30 seconds, you'll have had a full adrenaline response, and every target cell in your body will be activated. Now, that activation also needs to happen really quickly. We can't take too long. But as we said, it also needs to be adaptable. So what goes on inside the cell? Here in this diagram, we have the adrenaline shown as this little orange box and the receptor shown in blue. And I've actually shown 2 different pathways that can be going on here to show what might be happening. Now, the receptor itself, when it gets activated, it recruits this G protein, and the G protein is what does most of the work in the cell, or what sets off the rest of the work inside the cell. And this is where the adaptability comes in, because whilst adrenaline is always the same, and the receptor is essentially always the same, the G protein connected to it can vary. And it will vary based on slight changes in the very end region of this receptor. So one cell might have this protein called G1, and the second one might have G2. Now G1 will then go on to activate all of these other proteins. So in this case, it's a protein that causes a burst of IP3. What that basically means is there's a protein, this green protein here, acts as an enzyme which cuts the ends off of these phospholipids on the inside layer of the cell membrane, and those cut off phospholipids will then act as a signal which release this huge burst of calcium. Now this enzyme works really quickly to cut off these IP3 ends, and the IP3 ends very almost instantaneously cause this massive burst of calcium, and that means that you really rapidly get a huge response within the cell. And what does calcium do? Well, anyone who's brushed up on their A-level muscles will know. Calcium is essential for muscle contraction. So when we get muscle contraction here, it's actually smooth muscle, which is the muscle we find in our blood vessels. So if we're getting, it forms a ring around the outside of our blood vessels. So if we have a ring of muscle there and it contracts, we get a smaller diameter and less blood flow. So this is what will be happening for all of the veins and arteries, going to the non-essential parts of our body, like the gut. And that's how we get less blood supply to the gut. At the same time, for all of the arteries that lead to our important muscles, we might get something like the G2 protein. Now what G2 does is it has another really rapidly acting enzyme which produces this molecule called cyclic AMP, or CAMP. Now CAMP goes on to do a whole range of other things, but basically it does the exact opposite. It makes smooth muscle relax. So now rather than getting less blood flow, it expands and we can get more blood reaching the important bits of our body. And at the same time, it breaks glycogen down into glucose. Glycogen is the inert store of energy. Glucose is the stuff that we can actually use. So we're getting more oxygen and more sugar to our muscles. So adrenaline is a really good example of whole body signaling, which is what hormones do. But actually there's a lot of times when cells need to communicate on a much smaller, more regional level. So for example, as developing embryos, we need to be able to decide where structures like limbs or fingers might grow. And for that, we use things called Hox genes. Hox genes are actually quite like hormones, but rather than going through the blood, they just get produced in one area and then they diffuse out evenly. And that means you'll have a higher concentration at the center and a lower concentration around the edges. So at the center of one region of Hox genes, you might get a structure developing, like a limb or a finger. And in the regions which are less concentrated in terms of Hox genes, you might get supporting structures like a joint. Or with our fingers, the gaps in between each finger, you might get completely different signaling, like apoptosis, cell death, which is how we get the gaps between our fingers. And then again, when we look even closer, there's actually a lot of structures within even those regions that need to be able to organize themselves properly. Tiny structures like capillaries, which are the smallest blood vessels we have, or neurons, which are obviously just one cell thick, but are surrounded by other cells. So the neurons in our muscles, for example, will be surrounded entirely by other kinds of cells, but they still need to be able to organize themselves properly, which raises the question, how can we build a body and make tiny groups of cells specialized to do essential jobs, like being neurons or blood vessels, when they receive the same large area signals from proteins like Hox genes? So again, take a minute and pause the video, have a think. Okay, well this time we're just going to go through the one answer, which is a really ingenious method called contact signaling. So all the cells in one region are going to be getting the same large-scale signals from hormones and Hox genes, but when you need to organize structures on a single cell level, you'll just use single cells. So what happens here is, in this diagram we're seeing two cells, cell number 1 in the top and cell number 2 along the bottom. So how contact signaling works is something like this, and there are a few examples of this. But this is the mechanism called notch signaling, named after this protein notch, that actually, has a role in organizing capillaries and neurons. So one cell will start displaying this protein called delta, and another cell will start displaying this protein called notch. And when these two proteins come together, they bind, just like adrenaline did with its receptor. But now, when they bind, rather than activating a signaling system inside one cell, what actually happens is the big, long notch protein actually just gets cut. So all of the extracellular outside of the cell region of the protein gets cut off. And that means that the inside region can get snipped by a second protein. And this inside protein is now floating around inside cell number 2. And rather than just floating around aimlessly, it goes into the nucleus and is essential for turning on a whole set of new target genes. And so now this cell number 1 and cell number 2, which were getting the exact same signals and messages from their environment, are now getting very different signals because they've decided which cell is going to be which. So now this cell, with all of the notch signaling going on inside it, will end up specializing into one of those specific structures, maybe a neuron, maybe a capillary. It depends on a couple of other signals from its environment, what it will do. But okay, let's move on to another example, making move. Organisms from bacteria to humans. Everything that lives needs its cells to be able to move, maybe to find food if you're a bacteria, or in our immune system, they need to be able to move around to find infected cells or invading pathogens. So that raises the question of, these cells don't have eyes, they don't have any way of sensing anything other than what's at their exact boundary. How do they know which way to go? And if this, we're going to think about bacteria. So how can bacteria find food and move towards food when they actually have no way of comparing their food here to food over there? They only have their environment which they're in now. So again, take a minute, pause the video, and come up with any ideas. Okay, well, this is another really ingenious mechanism, which we call chemotaxis. So, since the bacteria can't look ahead to decide where the food is going to be best, they basically just have the propeller, which is how they move through their environment. And the propeller is actually called a flagellum. That's the technical name for it. And this propeller constantly turns off and on, and it pushes the bacteria forward when it does, but randomly it will just turn off. But the crucial thing is it stays on for longer as it gets more food. So in this bottom image, there's very little food in this white region, and there's a lot of food on a gradient in this green one. And as the bacteria goes along, from left to right, it will be getting more and more food, more sugars. And each time it moves into a region with more sugar, its propeller is able to keep moving the bacteria forwards for a bit longer. So if you look at this path, it's all random movement, but as it moves from left to right, it will stay on for longer than when it moves right to left. Moves quite far to the right and a very short distance left because the bacterial propeller just doesn't stay on as long. And so this is how bacteria can always move immediately towards their food, even though they don't know, in quotation marks, where it is. And okay, this will be our final example, exosome signaling, and we're going to switch it around this time. This is actually something that I research. This is the main focus of my PhD. And so, as an explanation, recently, in the last 15 years or so, it was discovered that cells can package large samples of their internal environment, so their proteins, their RNAs, lipids, sugars, and they can send them as signals to other cells. So the cells here are shown in red with a big blue nucleus, and the green tiny dots are the packaged exosomes, which contain bits of their internal environment. And crucially, all those molecules inside an exosome can be taken up by another cell. And that's really useful because rather than just using one molecule like adrenaline or a notch protein to convey a message, you can use thousands of molecules and give a really precise, complex message to another cell. So that can be really useful, but what I want you to think about this time is, now that we know what the signaling system is, what could be the risks of it if, say, there was one cell which had faulty or unhealthy signaling? So again, take a minute. Okay, well, maybe you got it, but basically you could risk spreading that disease or spreading that problematic signaling. So here on the left we have a diseased cell, and it's sending off a collection of its proteins, which are all potent signaling proteins. And when it gets inside another cell, well, with enough of these signals, you might be able to transform this cell into another diseased cell. And that's exactly what we see. So I mentioned at the start that I study how our cells signal in terms of cancer and neurodegenerative diseases. Well, these are two diseases where exosomes are really heavily involved. And what exosomes essentially let you do is transplant the processes going on inside of one cell into another. And that can be lethal if the original cell has something very wrong going on with it, like cancers. So cancerous cells can very easily corrupt their healthy neighbors using things like exosomes, and they can actually just make it much easier for themselves to spread, even if they don't directly change the cells around them with exosomes. And in Parkinson's disease, you get specific signaling molecules and unhealthy proteins called amyloid proteins. And it's been shown that exosomes can help transport these proteins between neurons much more efficiently than they would otherwise be able to. And that's how Parkinson's disease and many other neurodegenerative disorders are thought to spread between neurons so quickly. So in summary, signaling is everywhere, and it really does matter. There isn't a process in biology that I can think of that doesn't have some form of regulation going on by a cell signaling mechanism. But also, signaling mechanisms aren't just random, they have to match the function. So they need to be fast, or adaptable, or local, or body-wide. But all of those factors matter if you want to get the right signal and the right effect. And finally, many diseases, especially in Western populations where it's not so much infectious diseases, but it can be things like heart disease, cancers, neurodegeneration. A lot of those diseases can only really be understood by the signaling mechanisms going on and what goes wrong in those signaling mechanisms. So I hope you've enjoyed this springboard video on cell signaling. If you want to learn more about cell signaling and all the other processes going on inside our cells, then I really think you should. It can get really, really interesting. I've linked a few articles here. They're all from a website called Chemistry World, which I think strikes a good balance between detail and approachability for people who aren't already experts in a subject. And there's one podcast episode from that same website. Also, if you get the chance and there's a local library or your school library which houses these books, well, these are two books which I really enjoyed and actually helped me make the decision to come and study biology at university. Those are The Epigenetics Revolution by Nessa Carey and The Deeper Genome by John Parrington. But enjoy, and thank you for listening.